A good morning or afternoon to you. Thank you for joining us today. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members as well as the broader scientific community about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the adaptive immune receptor repertoire community, also called the AIRR community. Our speakers today, Susanna Marquez and Ken Hoehn, are active members of the AIRR community. Today, they'll tell us all about reconstruction and analysis of B-cell lineage trees from single-cell data using Immcantation. Please note the webinar is being recorded. Slides can be downloaded from the Materials tab in the viewer. Please do add any and all questions to the Q&A box in the viewer. The webinar does have two parts, and we will have a Q&A session after each part. Without further ado, I'll now turn the show over to Susanna Marquez.
Thank you, Jan, for the introduction. And thank you to the AIRR community and the Antibody Society for having us here today to share our work on airr-seq analysis. As you know from the title, Reconstruction and Analysis of B-cell lineage trees from single-cell data using Immcantation, we are going to show you how to create and analyze BCR lineage trees with the most recent methods in the Immcantation suite. Many of our lineage methods have been developed by Ken Hoehn as part of his research in immune repertoire phylogenetics. Ken is a postdoc at Steve Kleinstein lab at Yale University, and I am Susanna Marquez, a bioinformatician at the same lab. Noah Lee, a computational biology PhD student at the lab, contributed to the creation of this tutorial as well. This is an outline of everything we are going to cover. This session has been designed to be interactive, and during the first 10 minutes or so, I will introduce the Immcantation suite and will explain how to get ready to run the tutorial. If you can't follow along today, don't worry, because the code is available online and this session is being recorded, so you will be able to run the tutorial at your convenience. After this introduction, Ken will take over and will cover points 2 to 6. First, he's going to give you some background on B-cell phylogenetics. Then he will show you how to combine gene expression and BCR sequence data. He will also show you how to identify clonal clusters and reconstruct the unmutated germlines. Then he's going to move to building, visualizing, and analyzing BCR lineage trees. The Immcantation suite is a collection of open source Python and R packages that you can use to perform start-to-finish analysis of high-throughput adaptive immune receptor repertoire sequencing data. Immcantation is focused on B-cell repertoire analysis, but it can be used to work with T-cells. The individual packages are available through popular software repositories such as PyPy and CRAN, and the source code is also available from Bitbucket. The entire framework is also available as a Docker container, which includes third-party dependencies, example workflows, and reference data ready to be used. And all tools use the data standard developed by the AIRR community, which makes it easy to combine Immcantation with other tools that use the same data standard. For today's tutorial, these are the relevant Immcantation packages that we are going to be using. Alakazam, that provides tools to investigate diversity, gene usage, and amino acid properties. Shazam, that is used to perform advanced analysis of somatic hypermutation. Scoper, to identify clonally related sequences, and Dowser for everything philo, like building phylogenetic trees and test evolutionary hypotheses. Since Presto, the first package, was released in 2014, Immcantation has continued to adapt to new methods for repertoire sequencing and has released, on top of the software packages, extensive documentation and training materials, resulting in over 40,000 downloads in 2019. And Immcantation.org is the main portal, and you will find there links to all the sites of all the other tools in the suite. We have users all over the world, and they have very diverse profiles. We have users that are bioinformaticians that use Immcantation in their pipelines to analyze data, and we have other users that are software developers that incorporate Immcantation within their own software, like, for example, VDJ Server or VDJ Base and AR Galaxy. There is two main ways you can use Immcantation. On the left, having Docker installed, you can obtain the Immcantation Docker container, which comes with all the software tools, dependencies, databases ready. Or you can use a native installation. That means that you need to install all the software tools yourself, as well as the dependencies, the databases. Here on the right, you have the commands that you can use to install some of the tools that we are going to be using today. And we don't have time now to go into installation details, but remember that you have extensive documentation at immcantation.org, where you will also find installation instructions for each package. And you always can use Immcantation at googlegroups.com if you need help. All members of the lab monitor this account, and the best person to answer your questions will get back to you. And this is the most important slide for today's session. You should have received already an email with these instructions, and I believe we sent that email yesterday. You can also check the chat, because I think you will find the link there posted in the chat later. The easiest recommended way to follow along today's tutorial is to use a Binder, which is a website that we have prepared with a training container that has example data and a Jupyter notebook with all the commands that Ken is going to be using today. And you can easily access this container by visiting the Launch Binder link shown here in the upper left corner. And you should have received this link in the email I mentioned before. And once the site is loaded, you just need to open the Jupyter notebook with the name Dowser tutorial. And there are other options to run this tutorial. Another one is shown here. And this is if you want to download this training container, the same one that is a Binder. You basically need to use these commands to pull and run the container. And the run command will map the current directory in your machine into the results folder inside the container, and will also start a Jupyter notebook in the container. And you will see, you will get a message that says visit a URL. You just need to open the URL in your browser, and you will see the notebooks. And you could also use a release container, or if you have already the tools installed, you could use your local installation. But as I said, for today, we are going to focus on Binder because in the interest of time, we cannot provide today support to all possible technical issues for people using Mac, Windows, Linux, and Binder makes things easier. But if you want to try any of the other methods later and you need help, you can reach us at immcantation@googlegroups.com and we will help you. And that's it. Just visit the link that I mentioned before that you have here and in the chat. And do it now, because sometimes it takes a while for it to load, and you can have it loading in the background while Ken continues with the presentation. Once the tutorial, the website is ready, you need to open the Dowser tutorial file, and you will see the code that we are going to be using there. If you have trouble loading the site, don't let the technical issues prevent you from following the presentation, right? Because everything is recorded, and you can run the example and the code later. So just focus on Ken's presentation if you have technical issues. And now, Ken, the stage is yours.
Okay.
I will take questions if someone has questions, actually.
Cool. Fantastic. Thanks, Susanna. So if you have questions throughout, I think you can ask in the Q&A, and Susanna can monitor that. So before the analysis part of the tutorial, I'm just going to do a bit of review to make sure everyone's on the same page. So the cell population that this tutorial is entirely focused on is B cells. These are important for the adaptive immune system because B cells are the cells that produce antibodies. Antibodies, of course, are proteins released into the blood and mucosa that neutralize pathogens by binding to structures on their surfaces. If things go wrong, however, they can also cause types of autoimmune disease by binding to self-antigens, and they can cause allergies by binding to things like food antigens. Antibody structures are initially expressed as receptors on the surfaces of B cells called, creatively, B cell receptors, or BCRs. So by studying B cells and B cell receptors, we are studying the population of cells that produces antibodies. BCRs, in contrast to a lot of other cell types in the body, are highly diverse and are produced through a two-stage process. The first of which is VDJ recombination, in which gene segments are randomly spliced together. So the immunoglobulin heavy chain locus is composed of multiple V, D, and J gene segments. And when a new naive B cell is produced, one of each of these segments is chosen and spliced together to give a permanently rearranged genomic locus within the B cell. This is then transcribed, translated, and expressed on the surface of the B cell. Importantly, this new BCR represents a kind of stochastic guess in antibody space. This process repeats for each new B cell, so that each new B cell produces a more or less unique BCR. This produces a vast repertoire of naive B cells expressing different random guesses in antibody space. So of the around 10 to the 11 B cells in the body, there's more than 10 to the 14 possible receptor combinations. So this is a huge repertoire of naive B cells waiting to be bound to some kind of antigen. This is all well and good, but what we really want in an immune response is not random guesses in antibody space, but high affinity antibodies targeted to the threats that we're exposed to. And to get to that, there's a secondary process of BCR modification called affinity maturation. To modify B cell receptor structures, B cells express enzymes that damage their DNA at the BCR locus and introduce mutations at a rate multiple orders of magnitude higher than the background rate of mutation. These mutations can change the structure of the BCR, potentially altering its binding affinity. B cells with higher affinities survive more readily than those with low affinity, so they're selected. And over time, this produces high affinity antibodies, or B cells that produce high affinity antibodies, which can then differentiate into plasma cells that go to the bone marrow or memory B cells that go to the blood. This talk focuses on B cell phylogenetics, and the goal of B cell phylogenetics is to use the cells that we can observe, which are usually not the ones actively undergoing affinity maturation, and try to make inferences about the history of B cell lineages. So the way B cell phylogenetics works is that, say we have affinity maturation occurring as shown on the left. It's this process of cellular division, mutation, and selection. If we were to sample this process, so the sampled cells in this diagram are the colored cells, and then sequence the BCRs of the cells that we sample and compare the sequences to the clonal germline, we would see not a random scattering of mutations, but a hierarchy of mutations, some of which are shared among all cells, some of which are shared among only a couple of cells, and some of which are unique to individual cells. And we can use this hierarchy of mutations to infer a phylogenetic tree of this B cell lineage, in which the tips represent our observed B cell sequences, and the branches represent the pattern of shared mutations leading from the germline to the sequences that we observe. So if you compare this tree on the right to the somatic hypermutation process on the left, you can see it more or less recapitulates the topology. So there are many reasons why B cell lineage analysis is so useful, but one of the most important is the fact that B cell lineage trees provide a way to link different sources of B cell diversity. For instance, if you have data from biopsies and lineage trees contain data from those different tissues, then lineage trees can tell you about immunological connectivity between these tissues. If you have data of B cells from multiple cell types, and you have B cell lineage trees that contain multiple cell types, then they can tell you something about the history of cell type transitions. And finally, if you have B cell lineage trees that are sampled at multiple time points, then B cell lineage trees can tell you about persistence or reactivation of B cell lineages and possibly their evolution over time. So for this tutorial, we're mainly going to focus on cell types. because we're going to be working with single-cell RNA-seq data, but we have tools for analyzing all of these different data types. So the type of data we use to study this evolutionary process in B cells is called adaptive immune receptor repertoire sequencing, or AIR sequencing, which basically means sequencing the VDJ regions of B cells and T cells if we happen to be studying T cells. The reason single-cell sequencing has the potential to advance this field so much is that it allows us to sequence both the heavy chain and the light chain of individual B cells, and to pair that information with gene expression information from single-cell RNA-seq, which is shown on the right. This allows us to get a snapshot of B-cell genetic diversity of particular B-cell subsets within a sample, which is a very powerful way of studying immune responses. So let's get started with the actual tutorial. So first of all, everything in this tutorial is going to happen in the directory of the example data that we sent out earlier. So if you want to follow along, be sure that's unpacked and move inside of it. If you have the Jupyter notebook, then it should all be loaded. Inside, once you're into an R session or your Jupyter session, there are two data files. The first is a BCR TSV file, which we can read in using the alakazam function read_changeO_db. And the second is a Seurat object, which we can read in using the read_RDS function. So these data are all 10x single-cell RNA sequence plus BCR sequences. The way this technology works is that single cells are immersed in oil droplets where their mRNA combines with molecular barcodes that allow you to match BCRs and other mRNA sequences to a single cell. These particular data are a subsample of the data from a recent study our group performed with Ali Elabedi's lab at the University of Washington. And it consists of blood and lymph node samples taken from a single patient at multiple time points following influenza vaccination. So do check out this paper if you're interested in the data more closely. Also be aware what we're dealing with is a subsample of this data and not the full data set. So a bit more detail on these data objects. We are not starting this tutorial straight from the raw data. So from the Seurat object, cells have already been put through quality control pipelines and annotated by cell type. So if you would like more info on how to do that, check out the tutorials on the Seurat site when we added a link here. We can access these cell type annotations from the gene expression object using the idents function. So if we print out the first cell identity, we can see the name of the cell barcode here and the cell type annotation, which we can see is a CD4 T cell. So not every cell in this data set is a B cell, which is pretty representative of single cell RNA sequence data. The BCR table is in a format called Adaptive Immune Receptor Repertoire format, which is a community-sourced format that the AIR community put together and is designed to allow interoperability between different tools. So most BCR sequence data is in this format. These sequences have already been aligned and processed to the IMGT reference database, which is a database of human, VD, and J sequences. For info on how to do that, you can check out our various Immcantation tutorials. So we have a few different ways of doing that. They're detailed there. The data in this object really is just a big table. So each row is a sequence, and each column contains information about that sequence. These columns are important metadata, such as the cell ID, the vCall, the jCall, the sample, and the day. So if you want to look closely at the information we have for each of the sequences, you can use these commands. So since both data sets have already been processed, the first task is to consolidate the data from both into a common object. We can do this in two ways. We can either bring the BCR data into the Seurat object or take the gene expression data into the BCR object. We're going to do both, and we're going to first import the data into the Seurat object by matching cell IDs. So because of how this data was processed, these cell IDs are not identical between the objects. This is a common issue that happens in a lot of these types of analyses. We can fix this in the first two lines. We add the sample information to the BCR cell ID. So we just paste the sample ID to the cell ID in the BCR database. And we also remove the trailing -1 from the BCR cell ID. Be aware that this step will probably be different if you're analyzing a different data set. It may also be unnecessary, but it's always important to make sure that your cell IDs are formatted between these two different data types. So if we look at the first cell in our BCR database and the first cell in our gene expression database, we can see that they're more or less formatted the same way. They're just in different orders. So this cell is clearly a different cell from this one. To match the rows together, we'll need to use the match function. What this will do is locate the index of each row in the BCR data frame and match it to the cell ID of the gene expression object. So I have an example of how that works on the right. So if we have cell A, B, and C in our gene expression database, and cell X, A, and B in our BCR object, the first index of this function would be 2, because cell A in our gene expression database matches to the second row of the BCR database. The next value would be 3, because cell B in our gene expression database matches to the third row of our BCR database. And the last row would be NA, because cell C in our gene expression database is not located in our BCR database. Because of this, we can tell the proportion of cells that don't match to the BCR database by taking the proportion of match indices that are NA. And we can see here that is about 25%, which is actually pretty low considering there are a lot of other cell types in our database besides B cells. And also, if you're paranoid about these sorts of things like I am, you can check just to make sure that the cell IDs in your gene expression database all match to your BCR gene expression database using the match indices. And we can see here they do. So everything looks good there. There are a couple of ways to incorporate BCR data into a Seurat object, but the easiest is to just add a vector that tells whether the BCR is found in a cell. And for that, we just say gene expression database contains a BCR if the match index is not NA. Once we've done that, we can then plot a UMAP of all the cells that we've profiled and color those cells by whether or not they contain a BCR. So as you can see from this UMAP, the BCR cell types are the ones that are expressing BCRs. So our naive B cells, our resting memory B cells, our plasmablasts, and our germinal center B cells all have cells that contain BCRs, and the T cells, natural killer cells, and dendritic cells and monocytes don't express BCRs. So this is a great confirmation that the cells you identified by gene expression information are really B cells. At this stage, I would advise counting your blessings, because this does not always turn out like this. And if you find lots of T cells that are expressing BCRs or monocytes that are expressing BCRs, you might want to go through and double check the other parts of your pipeline. So this is looking good for now. The other way of combining these information sources, and for us a much more useful way, is to add gene expression information to our BCR object. It's very much the same process, but the match index goes in the other way. So if we go back to our same example and we're now matching from the BCR database to the gene expression database, the index is going to be first NA, because cell X is not found in our gene expression database, then 1, because cell A is in the first index in our gene expression database, and then 2, because cell B in our BCR database is found in the second row of our gene expression database. Similarly, we can check the proportion of BCRs that don't match to our gene expression database by just seeing what proportion of our match indices are NA. And we can see that's around 10%, which isn't bad. Sometimes this can happen if we sequence a BCR, but the cell associated with it is filtered out in other parts of the cell ranger pipeline due to QC filters. We can then use the following lines of code to add new columns to the BCR data frame. So these are columns that would contain the cell type annotation, the UMAP coordinates, and we can also filter out cells from the BCR database that don't match to our gene expression object. That isn't strictly necessary, but it cleans up the analyses later on. So now that we have our data, so because this is just a data frame with the BCR specific metadata columns, we can color the UMAP however we like. For instance, here we plot the same cells as in our UMAP, but we color them by our gene expression annotation. So we can set up a palette using each of our cell types that we've annotated, so germinal center B cells, plasmablasts, ABC cells, and then color the cells using those cell types using ggplot, shown here. As we can see, because we're just using the UMAP coordinates as the X and Y coordinates of our ggplot, this is very much the same shape as our total UMAP that we plotted using Seurat. Of course, it is only the cells that contain BCRs. So we can also color our cells by any other type of information that is within our database. So here we've colored them by isotype. And we can do that by just changing the ggplot call to specify color by isotype. And here we can see our UMAP, where each of these BCRs are colored. We can see that our naive B cells are mostly IgM. The cluster that represented plasmablasts are almost entirely IgG. And the other cell types are kind of a mixture, which is about what we would expect. So now that we've read our data into one session and combined it into an object, we can start moving towards building lineage trees. Before we can do that, however, we have to identify clonal clusters of B cells. Biologically, a B cell clone is a set of B cells that descend from a common VDJ arrangement. Or you could imagine them as descending from a common naive B cell. Importantly, these cells within a clone can differ by mutations due to somatic hypermutation. So for us, defining clones is particularly important because phylogenetics is all about descent from a common ancestor. So it only really makes sense to build phylogenetic trees within B cell clones and not between B cell clones. There are a few different algorithms available for grouping sequences into clones, but today we're just going to talk about our group's algorithm for single linkage hierarchical clustering. The way this works is we first group heavy chain sequences by common V and J genes and junction lengths. We then compare the junctions of each sequence within each group to its nearest neighbor in sequence space, and then pick a Hamming distance threshold based on these distances. We can then group sequences together via single linkage clustering. If you're interested in where these methods come from, the papers listed on the bottom left detail the development of these methods and validation of these methods. So the full references are in the notebook. They basically show different ways of clustering B cells into clones and how accurately we can define them, which is quite accurate. So the first part of this step is to define a Hamming distance threshold. We're going to do that using the Shazam package, which is also part of Immcantation. First, we use the dist to nearest function on our data. So this will group sequences by common V and J segments and junction lengths, and then calculate the distance of each sequence to its nearest neighbor within these V, J, and junction length groups. So it's important that we only use heavy chains for this step because we only want to be comparing like sequences with other like sequences. In this tutorial data, of course, we only have heavy chains, but in real data, there would be light chains as well. So I've included this filtering step just to avoid confusion. Next, we can use the find threshold function, which will look at the distribution of distance to nearest neighbor distances and automatically detect an optimal threshold. So the way this works is when we plot the distribution of distance to nearest neighbor differences, we often get this bimodal distribution. So the left mode is interpreted as comparisons within clones, while the right distribution is interpreted as comparisons between clones. And the optimal cutoff is the local minimum between them. So find threshold is really trying to find this local minimum between these two bimodal distributions. If you don't have a bimodal distribution, we have methods for grouping sequences into clonal clusters without that. But oftentimes, if you're dealing with B-cell repertoires, especially from blood samples, you end up with a bimodal distribution, and it's easy to find a threshold. So as you can see, the threshold we end up finding is just over 0.2, and we can create these plots using the plot density threshold function. Now that we have our threshold, it's fairly easy to identify clones. So we can pass our data into the hierarchical clones function in Scoper and specify the distance threshold that we determined from the previous slide. Once we've assigned clones through Scoper, we can use functions through dplyr or the count clones function in Alakazam to get a distribution of clone sizes. Once we have that, we can plot the distribution of clone sizes using ggplot. And this will show the number of sequences per clone across all of the clones within our data set. So as we can see, most of the clones within our data set have only a few sequences. There are a few with up to 20 sequences. So these are going to be the larger clones that we're going to build trees from. But this is a mostly typical distribution of clone sizes that we see from blood repertoires. The one exception is that normally within B-cell repertoires, there are far more clones that have only one sequence. As you can see here, there are a pretty small proportion of clones with only one sequence. This is because we removed those from this tutorial to make the data set a little bit smaller. But be aware that when you're analyzing your own data, you'll often find far more singleton clones than in this data set. Also keep in mind that when you run this command, or the hierarchical clones in Scoper, because we only have heavy chain sequences, you'll get a warning that Scoper is running in bulk mode. This is because when you're analyzing bulk data, typically you only have heavy chain sequences, and Scoper will assign clones based solely on the heavy chains. If you input single cell data with heavy chains and light chains into Scoper functions, it will run in single cell mode, and you'll have the option to use light chain sequence information to further define the clones. We're not going to get into that within this tutorial, but there is documentation about how to do that on the Immcantation site. So another important step before we start building trees is to reconstruct the clonal germline sequence. So this is the sequence of the unmutated naive B-cell ancestor for the clonal lineages. So on the right-hand side, we have the B-cell clone that we've identified, and the clonal germline is the sequence of the cell that gave rise to this B-cell clone. And we may or may not have sequenced that cell. Generally, we assume that we have it. So in order to reconstruct this sequence, we need some outside information. Here, that outside information is going to be the IMGT reference database. So this is a database of human and other organism germline, immunoglobulin, heavy chain, and light chain alleles. The easiest way to get this is to clone the Immcantation repository, which you can use using Git. So I should say these commands up here in this top left box should not be entered into an R session, but into a command prompt. So if you run this command, it will download the Immcantation repository, which has a set of scripts, one of which will download the IMGT gene DV reference database and format it in a way that's easy for Immcantation to use. So that's what these commands up here will do. Once you've done that, you can go back to your R session and read in the IMGT reference database using functions in our R package dowser. The function for doing that is read IMGT, and all you have to do is input the location of the IMGT reference database that you downloaded. So here we're analyzing human data, so the location, if you downloaded this database on your own, is human/VDJ. The location of the database, if you are using the Docker container, is this, the read IMGT user local share germlines. So depending on which way you're following along, just input either of these options. And this will create a list of all of the relevant IMGT reference sequences. From here, we just need to run the create germlines function with your data and the reference sequences. This will reconstruct the clonal germline sequence for each of your B-cell clones. And once we've run it, we can see here part of the first germline sequence within our database. We can see conceptually what this function is doing on the bottom right. So what this function is doing is taking the data from the sequence alignment column, which is shown the top row, and then taking the germline V, D, and J allele that matches to that sequence alignment and stitching it together to give an aligned germline V and J sequence. We also mask the D sequence and the junction region of the germline because these sequences are typically difficult to infer accurately. So we mask them with N nucleotides, which are ambiguous nucleotides. So finally, we need to do one last step before building our trees, and that is to format our data appropriately. So currently, our data set is still one big data frame where each row is a sequence and each column is information about that sequence. For building trees, we need to reformat this data into a different data frame where each row represents a clone and each column is information about that clone. So to do that, we use the format clones function from Dowser. This function wraps up a lot of tedious processing into one function, so there are a lot of options and they can have an impact in the tree that you end up building. So here we say that we want to specify the day, the isotype, and the gene expression annotation information as traits of the sequences. And we want to have the duplicate counts as the numeric fields of the sequences. So the way this works is format clones by default will collapse identical sequences together. However, if identical sequences differ by a trait value, which here we're specifying as the day, the isotype, or the cell annotation, those sequences will be kept distinct. Numerical fields will be added together if the sequence they're associated with is collapsed with another identical sequence. The minimum sequence field that we specify for format clones does pretty much exactly what it sounds like. It filters out clones that have fewer than the specified number of sequences. So after running this command, we'll have a new data frame where each row is a clone ID, and we have an associated data object called an airr-clone object. We also have information about the locus that's present, as well as the number of sequences within the clone. Now, hopefully, you'll never have to deal with one of these airr-clone objects directly, but just to show you what's going on under the hood, we can look at what is inside of an airr-clone object. And we can see that it's just one big data frame that has the sequence ID, the sequence information itself, as well as the metadata columns that we specify in format clones, such as day, isotype, expression, annotation, and the number of sequences that were collapsed to this identical sequence. It also contains some information about the clone, such as the V gene, the J gene, the junction length, and other information such as the IMGT numbering. So we're finally ready to start building trees, right? Sort of. Turns out there are many different ways of building phylogenetic trees from B-cell sequences. The oldest and still quite commonly used is maximum parsimony. So this works by finding the tree that minimizes the number of mutations present within the B-cell lineage. The benefits of this are that it is fast, simple, and it's easy to interpret. The downsides is that there are some instances where maximum parsimony can be positively misleading, such as when parallel mutations are likely. So in those cases, you can very consistently get the wrong tree topology. Example programs for doing this are DNA parse from the phylum package and Fangorn in the R environment. Another option is maximum likelihood, which I'm going to call standard maximum likelihood. The way these work is by defining a Markov model of mutation rates. And we find the parameters and tree topology and branch links that maximize the likelihood of observing our data. The benefits of this is that it can be less biased than parsimony in some cases. The downsides are that it can be a little more computationally intensive and also sensitive to poor model choice. There are a lot of example programs for this. One of them is DNAML, which is also distributed as phylum, but also RAXML, PhyML, Codon PhyML, and also methods within Fangorn, which is in the R environment. And finally, there are options for B-cell specific maximum likelihood models. So these are Markov models that incorporate B-cell specific mutation biases. These have the benefit of reducing parameter estimate or bias in parameter estimates in your data, and in cases where those parameters are important, like if you're trying to estimate the force of selection or the rate of different types of somatic hypermutation, this can be very useful. The downsides is that these methods can often be quite slow, and the main example program for that is IgPhyML. So fortunately, or perhaps unfortunately, if you're indecisive, is that you can build trees using all of these methods within the Dowser package. All you have to do is specify the getTrees function and change some of the options. So by default, we can just pass the clone data frame to the getTrees function, and Dowser will find the maximum parsimony trees for each of the clones. So this will use the R package Fangorn to find the set of the most parsimonious trees for each of the clones within our data frame. If you, however, want to use DNA parse, which is quite commonly used in BCR sequence analysis, all you have to do is specify DNA parse in the build field of the getTrees function and pass it the location of the DNA parse executable program. This will vary depending on your setup. This is the location of DNA parse in the Docker image. If you did a local installation, then you will need to pass the location of wherever you installed DNA parse. To build maximum likelihood trees, you can just specify PML in the getTrees function, and this will use the Fangorn package's maximum likelihood methods to build trees for your data. You can also specify which substitution model you want to use to build your trees. Here we're specifying the general time reversible model, but there are other options available. This is also the default if you don't want to mess around with the substitution model that's being used. If you would prefer to use DNAML, which is also commonly used in BCR analysis, again, you just specify the build option as DNAML and pass the location of the DNAML executable. And this will build trees using DNAML. Finally, if you want to be super cool, you can build trees using IgPhyML. Here, again, all you need to do is specify build IgPhyML and pass the location of the IgPhyML executable. So this is the executable location in the Docker image. If you've installed IgPhyML locally, you can specify that wherever you've installed it. I will say, as a bit of a caveat, this can be quite a computationally intensive step. So if you're following along with the Binder website, it's best to not run this on Binder, and give this a bit of time to run if you want to run it while following along. In either case, what we're going to end up with is a data frame of each of our clones. So this is the same data frame that we formatted before, except we now have a column called trees, which is a column of the phylo objects. So just like how these AIR clone objects contain information about our BCR sequences, these phylo objects contain information about the B cell trees that we've just built. So regardless of how you built the trees, they're visualized in the same way using the plot trees function, which uses this really great package called ggtree. And it allows you to plot phylogenetic trees like ggplot objects. To use this, we just pass the function plot trees, which is a function within Dowser that largely acts as a wrapper for some of the ggtree functions. We just pass the trees data frame to the plot trees function. We specify the tips as isotype. So this means we want to color the tips by the isotype of their cells. And we specify that we want a tip size of 2. So this just toggles how large we want the tips. If we want to plot the largest tree, we just plot the first element in this list. So this function will give us a list of plots. If you just enter the plots list and try to plot all of them in an R terminal, you'll end up plotting all of the trees sequentially, which is not a very useful thing. What you can instead do is save this list as a PDF file. So if you use the trees to PDF function and specify the file that you want to output and the number of rows and columns you want, it will make a nice PDF with all of the trees within your database with the specified number of trees per page. So this is the first page of what that output will look like when running these commands. Now, what I just showed is a set of fairly simple plots. In reality, publication quality figures are usually much more elaborate and require a bit of fine-tuning. Fortunately, it's easy to modify our plots once they're generated by using functions from ggtree and ggplot. So for instance, here we plot the second largest tree in our data set, and we're going to make a few changes from the previous plot. First, we're going to scale the branches of the trees so that the branch lengths represent the total number of mutations between the nodes of the tree rather than the mutations per site. So in this slide, you can see the scale bars here are quite low numbers, and these represent the number of mutations per nucleotide site within the tree. Here, the scale bar is a much higher value, and that's because we've rescaled the tree so that the branch lengths represent the total number of mutations between the nodes in the tree. We're also going to want to color the trees by the cell type annotation, and we're going to use the same color scheme that we defined in our UMAP. So if you look at the bottom left, we have the UMAP that we plotted earlier in the tutorial when we were just doing our gene expression analysis. Here we have the same cell types and the same colors on a phylogenetic tree inferred from this data. And we can see the plasmablasts, the germinal center cells, as well as a resting memory B cell, that is an IgA cell. We've also scaled the tips by the sample date, so we can see that these later sampled cells at the bottom were germinal center B cells and mostly IgG. We've also labeled the cells at the tips by their isotype. So this is quite a rich plot. There's a lot of information plotted on the tips of these trees, and this is an example of what you can do with using ggtree. You can really plot basically any kind of information that you can associate with a cell on the tips of the tree. It's just important that you when you're formatting your clone objects, specify that you want to store those columns within the clone objects. So a bit of a change in subject. One major application of B-cell lineage trees is to reconstruct sequences at the internal nodes of the tree. So doing this is easy in Dowser. In fact, Dowser will automatically reconstruct the sequences of each internal node of each tree automatically without any further specification. All we have to do is tell Dowser which sequence at which node we want. So to do this, we need to first process our trees using the collapse nodes function. This isn't strictly necessary, but what it will do is take internal nodes of the tree that have identical predicted sequences and collapse them into one node. This will clean up the plot of the tree significantly. Next, we make the same plot that we did before using exactly the same command, but we specify this option node nums equal true. And what this will do is plot the exact same tree, but it will label each of the internal nodes by the number of that node. So here, let's say we're interested in the most recent common ancestor of this lineage tree, which in this plot is node number 26. It may be different if you're running this program yourself, but the main thing to do is to look at this plot and identify the number of the node that you're interested in. From there, we can use the getSeq function, where we pass the trees object, we pass the ID of the clone we're interested in, and the number of the node that we're interested in. And we will we will print off the sequence of the heavy chain of that node. So importantly, this is a sequence that is not actually part of our data set, but is predicted to have existed based on this phylogenetic tree. And we can even see, if we look closely, some sequences that are not actually nucleotides. For instance, in the second line, there's a V, there's also an R. Those are ambiguous nucleotides that represent uncertainty in the ancestral sequence reconstruction. And those are important things to take into account because oftentimes there are multiple different ways of reconstructing the series of mutations along a B-cell lineage tree. Now that we've built and visualized our trees, I just want to reinforce the point that B-cell lineage tree analysis is a powerful tool for studying immune responses, in part because trees link different sources of B-cell diversity. My research in particular focuses on using B-cell lineage trees to understand immune responses, and I've developed multiple approaches for doing that. For instance, one way of linking B-cell diversity is to use data from different tissue biopsies, which can tell us about immunological connectivity, or data from different cell type transitions, which can tell us about how cell type transitions are occurring in B-cell lineages. And for doing those kinds of analyses, if you're interested, we have a preprint out detailing one method for analyzing these discrete data types in B-cell lineages. Another interesting data type is to use B-cell lineages that are sampled at multiple time points over time. And if you have lineages that occur at multiple time points, this can tell us about persistence or reactivation of B-cell lineages, and also evolution over time. If you're interested in that, we have a preprint detailing how we can use lineage trees to detect measurably evolving lineages. So just to give an example of how B-cell lineage trees can be used for these analyses, and to focus on one example that I've been especially interested in recently, I'm going to talk about how we can detect ongoing evolution in B-cell lineages using B-cell lineage trees in Dowser. So more specifically, I'm interested in whether B-cells are what's called a measurably evolving population. So these are populations that evolve rapidly enough for significant genetic differences to be detected in serially sampled time points. One example of a measurably evolving population would be the coronavirus. So if you were to sample the worldwide population of the coronavirus last year and compare it to sequences from this year, you would find loads of new genetic differences in the form of the variants that now have everyone quite concerned. Alternatively, if you were to sample a population of tortoises from last year and then this year, you probably wouldn't find very many genetic differences because tortoises evolve quite slowly. So we could say that the coronavirus is a measurably evolving population, while tortoises are probably not. Importantly, both of these populations are evolving. It's just only one of them is evolving rapidly enough to be measurably evolving. So using longitudinal samples from the same patient, we can determine whether B-cell lineages are actively evolving over the time interval using B-cell lineage trees. So take, for example, this lineage at the top left. This was sampled over a couple of different years, so it was obtained from a chronic HIV patient. So we can see the first sampled time points, which are colored dark blue, are very close to the germline of the tree, which is in the bottom left. So this indicates that the sequences that were first sampled were not very diverged from the germline, but the sequences sampled at later time points are very diverged from the germline. So this is exactly what we would expect from a measurably evolving population. One way of viewing that is to use a scatter plot where the x-axis is the sample time and the y-axis is the divergence from the germline. And if we do that, we can see a very clear increase over time within this particular lineage. And the slope here represents the rate at which mutations are accumulating for each week, because that's the unit of our sample times. Importantly, because these tips are not independent data points, it's improper to use standard linear regression p-values to estimate the significance of this relationship. What we can do instead is to use a permutation test to determine whether we have a stronger correlation in our observed data than in the same tree, but with the time points randomized at the tips. And here we can see that we very much do. Alternatively, if we look at this lineage at the bottom left, we can see there's an almost random relationship between the sample time and the divergence. And this is actually quite common in B-cell lineages, perhaps surprisingly so. If we look at the scatter plot, we can see there's very little relationship between sample time and divergence. And if we look at the permutations of correlation over time, we can see that our observed data is not significantly different from the same tree, but with randomized data. So performing this test is easy to use in Dowser and is implemented in the function correlation test. So all you need to do to run this is to input your data frame with your estimated tree topologies into the correlation test function and to specify which column in your data frame represents the numeric values for your sample time. You can also specify the number of permutations. By default, it's 1,000. For any kind of published analysis, I would recommend doing like 10,000 or even 100,000 permutations just to get a more stable estimate of the p-value. Then you can just remove the trees that have only one sample time. You can identify them because they have an NA value for their p-value. This makes sense because if you only sample from one time point, there's no correlation between the sample time and divergence. We can then order the trees by their p-value so that the most significant trees are at the top of our data frame. Then we can use our plotting functions to create really fancy looking plots where the trees are plotted using scaled branch lengths and the tips are colored by the sample day. And here we can see the top 4 trees that show the strongest signal of measurable evolution. And we see the top one is, it for sure shows a positive correlation between divergence and time. We can see the earliest sample time points are closer to the root than the latest sample time points. However, all of these trees are pretty small, so the p-values are not quite significant, and overall, it's not quite convincing that these lineages are measurably evolving. If we had a larger data set, we would probably be able to detect at least some amount of measurable evolution within this data. So just as a recap for this tutorial, the material we covered is first setting up Immcantation. Then we went into a bit of background into B-cell affinity maturation and phylogenetics. We covered combining gene expression information with BCR sequences, then identifying clonal clusters using BCR sequences, building and visualizing trees using Dowser and ggtree, and finally doing a small amount of tree analysis to detect measurable evolution of B-cell lineages over time. Just to be clear, everything in this tutorial is part of the Immcantation suite, which is a start-to-finish suite of programs for BCR analysis developed and maintained by the Kleinstein Lab. So if you're interested in using these tools, check out our website at immcantation.org. That is the main link, which links to all of our other documentation sites. We put a lot of effort into maintaining and documenting these tools to keep them working and user-friendly. We also maintain multiple beginners tutorials. So if you're looking to get started with Immcantation, there are a lot of resources on our site. This screenshot in the center is a sample of some of the vignettes we have for using Immcantation. So if there's something you want to do with BCR sequences, there's a good chance you can do it in Immcantation, and that we've listed at least something on our website for how to do it. So with that, here are the links to the documentation for the most relevant software, including the Immcantation portal itself, Dowser, which is the phylogenetics package that we've recently developed and was used for building trees in this tutorial. IGPhyML, which is a phylogenetics package for building lineage trees using B-cell specific substitution models. If you have any questions you want to ask me directly, feel free to email me at this email address. If you have any questions about actually using Immcantation, please email this immcantation@googlegroups.com list. As Suzanne mentioned, this goes to our lab, and the person best suited to answer your question, which may very well not be me, will reply. So your best bet for getting software help is to email this immcantation@googlegroups.com list. If you're interested in B-cell phylogenetics more generally, I've listed some relevant papers here. So the first is on the bottom left, a review article on B-cell phylogenetics and some of the challenges and potential for it. Just to the right of that is the paper detailing the models that go into IGPhyML and how it adjusts for the unique features of B-cell affinity maturation. Above that is our tools for analyzing discrete character traits such as tissue location, cell state information, and class switching. And just above that is our methods for detecting ongoing B-cell evolution from B-cell lineages. Also, I really want to give a big shout out to Noah Lee, who is a grad student in our group who did a lot of work in putting together this tutorial, as well as Hailong Meng, who is a bioinformatician in our group who helped put together the tutorial data for this tutorial, as well as some of the earlier parts on matching B-cell receptor information to 10x data. And of course, our PI, Steve Kleinstein, who supports and supervises all of this. Finally, in part because I'm currently looking for faculty positions, there are openings in the Kleinstein Lab. So if hearing all of this sounds super cool and it's something you would be interested in doing as a full-time job, we have open positions for postdocs and bioinformaticians interested in computational and systems immunology. So we have really great experimental and clinical collaborators working with single-cell RNA-seq data, as well as BCR data and TCR data, multiomics, and a lot more. So if you're not just interested in B cells, there's plenty of stuff in the Kleinstein Lab for that. We also do a lot of things for developing visualizations and notebooks to support analytical pipelines. So if you're interested in that, please email Steve at this address. And with that, I can take questions. I guess I'll check the Q&A. Okay,
Hello.
there are a lot of
Yes, there are a lot of questions. So that's good. That actually caused a little bit of a delay on this side, looking at all of these as to whether we would actually have time to go through them all. I would ask then maybe for you to look through the questions and pick the most relevant ones where you feel the ones that you could answer relatively quickly. And then what we can do for the attendees is to take the questions and have you answer them and post them as a Q&A document that's also sent out to the people who registered. So I will ask you to give that a second's consideration and perhaps pick a few questions that you think.
I can pick one because I prepared a link for that one already. It's the one, how much improvement do we get on the B-cell clonal clustering by adding the light chain? And there is a little improvement, but not much. We have methods that subdivide the clustering using also the light chain information. But we have a paper from the lab that I'm going to put the link here, if I figure out how. Okay, yes, here. Where we show that there is not much improvement, that just the heavy chain is enough to do the clustering.
Yes. So I guess I think some of these, a lot of these I could probably answer relatively quickly. Should we just start at the top and go down, or?
Sure. If you want to take 5 minutes or so.
Okay. How much time do we
aim to end at 1:15?
Okay. Cool. Or are these okay? I guess these were asked in reverse order, so maybe. Okay, so Susanna asked that. If we are starting with gene expression data that hasn't been annotated by cell type, how could we generate these annotations? So there is a link in one of these slides to the Seurat site, and they have tutorials for that. So yeah, if you look at slide 16 and just follow the link there, they have tutorials for using tools in Seurat for annotating cell types. How to incorporate light chains to cluster B cells. So if you go to the website Scoper, so yeah, so if you look at just Google Scoper Immcantation or go to the Immcantation site and select the Scoper icon, it will take you to the documentation site. And there are some, there's some detail in the package for how the light chain clustering is done. Basically, you infer the clones using the heavy chain information and then split them into smaller clones based on whether the light chain B and J genes are different. So the threshold is always automatically picked. Not always. Sometimes you can have a threshold or a distance to nearest distribution that looks bimodal enough, but the automatic threshold detection doesn't actually work. And sometimes you can just pick the local minimum manually. We also have methods for what's called spectral clustering, which don't require a fixed threshold, and that is also detailed on the Scoper site. Can we select heavy and light chain at once to compute the threshold? So currently the Hamming distance threshold detection tools all use the heavy chain. So when you're doing those, it's important to only give it heavy chain sequences. The light chain information, if you're splitting clones by those, comes in at a later step. Okay, I'm using 10x single-cell genomics and later using FASTA annotation to run IG-BLAST sequencing alignment, but I know the Cell Ranger pipeline doesn't arrange all of the BDJ BCR sequences correctly. So how do we rearrange it so it is correct, and would you recommend quality control on the BCR data before classifying clonotypes? I don't fully understand the question. So we have a tutorial on our website for doing basic 10x genomics BCR sequence analysis. It starts at a slightly earlier stage in the pipeline. So let me find where that is.
In the answer, Ken, already.
Oh, okay.
While you were presenting, I was trying to collect information to help with the answers. Okay,
cool. Yeah, if you have an answer for one of these, please chime in as well. But I can. Yeah, that's helpful to just have the link. Can heavy and light chains at once compute the threshold? I think we answered that earlier. Are there tutorials using Immcantation tools for B-cell lineage construction for bulk BCR sequences? Yes. So bulk analysis is almost exactly the same as this, but you don't have light chains. So in a way, we kind of already did bulk sequence analysis, but on our website, there should be tutorials for how to use that. If you go to the Dowser website, it has vignettes walking through a data set that is essentially all heavy chains, so it's basically bulk sequencing. So for bulk, you'll probably have more luck looking at the change-o documentation. So will it be possible to use paired heavy and light chain sequences to run IG-FIML and build a tree based on both sequences? Yes, so that is something that is in the pipeline. Hopefully will be coming out soon. If you're really interested in that, send me an email and I can talk to you about how to do that with the tools we currently have. But yeah, one thing we are very much looking to do in the near future is incorporate heavy and light chains into building B-cell lineage trees, including an IG-FIML. Can clonal lineages account for correct class switching events in lineages constructed for BCRs? So we don't incorporate isotype switching events into the tree inference, and I don't think it's generally a good idea to do that because the trees represent mutations, and there isn't necessarily a one-to-one relationship between the mutation process and the class switching process. So we can reconstruct the pathway of likely switching events through a tree that incorporate the constraints of class switching events, but those aren't actually used in building the tree. Hope that answers the question. So that's actually detailed in the Dowser paper here. So in the middle right paper, phylogenetic analysis of migration, differentiation, and class switching in B cells, that details some of those algorithms for trying to reconstruct the series of class switching events along the tree. Final question, should we rely more on heavy chain because it's diversity in the light chain or use both? Lastly, we define clonotypes. If these clonotypes are real, should they be confined to a specific cluster of cells on a UMAP? So the first question, for most analyses use the focus on the heavy chain. And I think part of that is because it has more diversity. Part of it is because a lot of people are used to using bulk data, which usually only has the heavy chain. But you can use both. For clonotypes, most of the information is in the heavy chain. When we define clonotypes are real, should they be confined to a specific cluster of cells on a UMAP? No. So it's quite common to find B cells that are part of the same clonotype that come from different parts of the UMAP. So this...
Yeah, I think we're going to have to close out here.
Okay.
Sorry, there's a couple of other questions, and the speakers will get back to you with responses. I do apologize. We have a hard stop here. So I do want to conclude and thank our speakers for relating their insights into reconstruction and analysis of B-cell lineage trees from single-cell data using Immcantation. And thank our attendees, or thank you to the attendees for joining us today. There will be an on-demand version, and I will send you a link. Please do feel free to watch this or any of our on-demand webinars when it's convenient. And thanks again. Have a good rest of your day.
Thank you.